This is a production of Cornell University.
All right, well, it's such a nice spring day out there, and so I want to reward you for staying inside with me with some beautiful spring flowers. I'm going to talk about a rhododendron project that I've been working on. It's been a long-term project, but that's woody plants for you. They tend to be long-term. The title is just meant to convey my effort to really rejuvenate a plant which has been popular for a long time in gardens, but has sort of languished in commerce and sort of has lost popularity in the public imagination. It's not for lack of aesthetics. It's an incredibly beautiful plant, and every new variety that comes out seems more stunning than the one before. The difficulty, the problem really lies with the culture of the plant, its biology. It's very difficult to grow. Unless you're a knowledgeable gardener that's willing to make the effort to do so. So for really the vast majority of the gardening public, these are plants that fail in the garden. We call them, in our cynical moments, biennials. You buy them in bloom at the garden center, and they die the next year. Well, our research department at Holden consists of 4 principal investigators. So why this big investment in rhododendrons? Well, part of that is because of it's happenstance. It's a convergence of like-minded people with the same interests, able to pool their resources and create what became the David G. Leach Research Station. So David Leach was a influential American horticulturist, president of the American Rhododendron Society for many years, involved heavily with the Garden Conservancy, but also a passionate admirer of rhododendrons and a world authority, really. He wrote his seminal book called Rhododendrons of the World, which really came out in the early 60s, really helped popularize the plant in the American consciousness. And his niche as a hybridizer was cold hardy hybrids. So his target for hardiness was zone 5, and in ornamental plants, the hardiness is usually given for the buds, the flower buds. That's the trait of interest. So buds hardy to about minus 15 Fahrenheit was his threshold level. And he created these beautiful hybrids here, the red C and the yellow Capistrano, which are plants, they're recombinant plants. You don't find those colors in a hardy background in nature. They're created by hybridizing different species. The other answer to why rhododendrons is why not? They really are probably the most, one of the most ornamental woody shrubs available in terms of the diversity of colors. These are species, most of them from Asia, showing the range of colors, everything except maybe a blue, variety of types of inflorescences. Tremendous variability in leaf morphology. This is, you have low elevation tree species with leaves that are 3 feet long, up to small, tiny little, small-leaved alpine plants at about 18,000 feet in the Asian mountains. Oftentimes they'll have these kind of fuzzy undersides here, different colors. It's kind of a suede type covering called indiamentum. Don't really know the function of it, but it's highly ornamental. So what I've been showing you so far is just a slice of the total genus Rhododendron, which worldwide consists of about 1,000 species. And that includes other subgenera, which are such as the deciduous azaleas and the evergreen azaleas and the small-leafed rhododendrons. Subgenus Hymenanthes, which is the focus of my talk, are the non-scaly-leafed, large-leafed rhododendrons, most of them evergreen. And the center of diversity, center of origin for this genus is southwest China into the Himalayas. So those thawed and hatched areas of extreme topographical relief, which led to quite rapid speciation. And then outward from that center, you'll find instances of other species into Korea, Japan, Central Asia, and Europe, and across the land bridge into North America. So there are a couple species on the in the eastern mountains of North America that are very useful for breeding cold hardiness. So breeders like David would typically take a hardy North American species and cross it with a more colorful but less hardy Asian species. And as a rule, you can intercross quite freely at the subgeneric level. And so there's really you don't have to go to extremes to try for these wide crosses. And the offspring themselves are often fertile, so hybridizers would just cross species with species, and oftentimes some of the modern varieties have as many as a dozen different species in their background. Where you don't find rhododendrons is in these continental areas of Asia or the US, such as Cleveland. And that's the areas where of extreme heat and cold. Rhododendrons like higher elevations in the mountains or along maritime climates, which are more buffered in terms of temperature. All right, well, briefly, the interest in the West in rhododendrons really evolved out of that crazy Victorian passion for plants. A lot of the initial collections coming into the West were in England, sponsored by landed gentry who could fund expeditions, and in return they would get seed. So really, from the get-go, the interest was among hobbyist enthusiasts, and that has really continued into today's world. Most of the breeders are just what I call backyard breeders. I'm definitely an exception to the rule. And some of the first hybrids were made in England. The Earl of Carnarvon created some of the first rhododendron hybrids, and he was the owner of, it was called Highcrest Castle, which you know as Downton Abbey. There you go. All right, so a century and a half later, there are over 25,000 registered hybrids. Not uncommon in popular plants like roses. Let's see here. Okay. The style of breeding was done as pedigree breeding, just like dogs and horses. Breeders had their favorite stud plants to use. Very limited trialing. Most things were introduced commercially based on one plant at one location. And the emphasis really was on flowers above all, and less on performance and adaptability. And that's been a problem in terms of general public use of these plants, particularly soil issues, the requirement for acid soils, pH optimum around 5, and very good drainage is required. And that primarily has to do with avoidance of root rot disease. And so many rhododendrons that are planted by gardeners who are not knowledgeable, buying them at big box stores and just sticking them into the foundation next to their downspouts, involves improper site selection and lack of proper soil amendment to try to overcome those restrictions. So really out of that 25,000 group of named hybrids, there's fewer than 20 that are important commercially on have a national sales presence. And those are the so-called ironclad rhododendrons, which were developed over a century ago in England, but have proven to be very versatile. And so my goal really is to try to expand that ornamental palette and create really the modern ironclads. So I'm taking a collection that David Leach created, really a 50-year body of work that he did that developed these highly ornamental cold hardy plants, and we're adding traits like root rot, disease resistance, and heat tolerance to those with a goal of getting a plant product that people can grow more successfully. Also one, particularly with the addition of heat tolerance, that will broaden the market. Rhododendrons typically fade out of culture as you go south around the Mason-Dixon line, and as you go south, the emphasis shifts to evergreen and deciduous azaleas. So rhododendrons are not considered to be particularly heat tolerant. And disease resistance and heat tolerance are not really coincidental traits. Our source of resistance that we're using happens also to be quite heat tolerant. And we feel that there's probably a functional relationship between these two. A working hypothesis in the breeding program is that disease resistance is important in warm climates because disease pressure increases as you get warmer and wetter. So the disease, root rot disease, is caused, the main causal organism is Phytophthora cinnamoni, which I will sometimes refer to as a fungus, but truly it's not a true fungus. It's fungus-like. It's now classified in the Protistas. It's a soil pathogen. When it finds a susceptible host, it colonizes right behind the root cap and then begins to grow through the root system, eventually moving up to the crown region. You can see this section here, the necrotic regions. At that point, it's really disrupting the vascular system of the plant. And you see this wilt symptom. That's the classic indication in the shoots. And that's irreversible. At that point, the plant's as good as dead. The zoospores are the infectious agents, shown here in the lower right. And they are motile. They have flagella. They can move a little bit around in the soil and under wet conditions. And so that's one of the reasons why drainage is so important as a disease avoidance mechanism. Well, Phytophthora is a problem for rhododendrons and many other crops because it's a global invasive. It's everywhere, and few native plants like many of our rhododendrons are resistant. So early on we were looking for sources of resistance, and when I started, there already was a report done by pathologists at Ohio State in the mid-'70s, and so we did another report in 2002 looking at more contemporary hybrids, just to do screens and identify either cultivars or species that would be useful for breeding. Only 6% of the hybrids proved resistant, and 17% of the species out of these collective studies. And that number gets even smaller when you look for plants that are not only resistant but also have some measure of cold hardiness, which is another one of our key criteria. So doing resistance screens these are done in controlled greenhouse experiments where we culture the pathogen in the lab and then make an inoculum and give it to the plants. So we're either doing these, like on the left, we're doing inoculations of single plants in pots. That's when we're usually trying to identify resistance level in specific genotypes where we need a replicated trial. Or we're looking at seedling populations, breeding populations, where we're screening genetically variable offspring, and these are done when the plants are at about 6 months of age. One of the key metrics that we use are disease root scores, or root rot disease scores, which is a numerical visual rating where on a scale of 1 to 5, so 1 is a healthy root, nice white fine roots here, working up to a level of intermediate level, which is 3, where you have necrotic fine roots plus some of these coarse roots here are also damaged. And we use pruners to kind of cut and inspect cross-section as we go. It's a destructive sample. Up into the crown rot area, and finally 5, which is a dead plant. So it's important for a parent to be resistant, but also for it to be able to transmit that resistance to offspring effectively. And not all plants do this equally well. It's based on really a breeding value, which in turn is a measure of heritability, the resemblance that offspring have to their parents. And this is a key thing in woody plants especially. You want a high heritability value, which is estimated on a scale of 0 to 1. because you want to make as much gain as possible in one generation. The generation time in woody plants can be long, and if you can get if you make good progress in one generation, that's really valuable. So based on these are the parent scores on the scale of 1 to 5 versus offspring on the same scale, and we're looking at open pollinated offspring of all these parents. And so the heritability estimate, which is narrow sense heritability, a measure of additive genetic variability, is 0.86, which is a satisfactorily high number. That just suggests that we can make pretty good gains in one breeding cycle. And the value comes in line with some heritability studies done on other woody plants. Mostly, these are all threatened tree species, where areas where Phytophthora is actually naturalized and is becoming an ecological issue. Eucalyptus, Monterey pine, European chestnut. So the values there vary from 0.5 up to about 0.86.
Do you have any similar data about prunus rootstocks in that respect?
Prunus, I don't. Ultimately, through some trial and error, we settled on one species as our foundation for resistance breeding. It's called Rhododendron hyperethrum. It's native to Taiwan, but its high enough elevation is found between 1,000 and 2,000 meters in the central mountains of Taiwan, so it has some hardiness. We can grow it. It's probably a zone 6 plant. We can grow it outside in Ohio. It did survive this winter, but it took a pretty hard hit. But its other virtue is heat tolerance. So this is one of the few large-leafed evergreen species that will grow in the Gulf South. In addition to those traits, it has good ornamental traits, this compact, bushy growth, glossy foliage, nice thick leaves, and it's very floriferous, although it only comes in white, and the inflorescences, or trusses as gardeners call them, are a little bit open. Typically the standard is for something a little more compact and organized. So from a breeding's perspective, what we're crossing this with plants that we know have hardiness, have good saturated colors, pinks, reds, yellows, purples, and so forth. So in a breeding experiments, and again, these are inoculated greenhouse studies, this just gives you a visual idea of what comes out of a cross. This is a susceptible cross. In fact, most of the parents we're crossing it with are susceptible. This is a variety called Calsap, open pollinated, so there's about 100% mortality. This is 7 weeks after inoculation. Crossed with the species, no mortality. And the F1 shown here, there are a few dead seedlings, but otherwise things look pretty good. So there's a strong resemblance of the first generation to the resistant parent. And you can quantify that just by doing disease progress plots. This is using another susceptible variety, yellow one called Capistrano. This is the mortality rate on the Y over days from inoculation. So this is the open pollinated offspring from Capistrano, a fast trajectory to extinction. R is the hyperethrum resistant parent. So it levels off at about 20% mortality among those seedlings. There seems to be some recombination where about 20% of the offspring are susceptible rather than resistant. And then the F1 plotted here, which levels off, and it tracks closely to the resistant parent. You measure the area under the disease progress curves and just subtract and divide, and you can end up with a gain of, in this case, 83% gain in resistance in one generation. So with some of those breeding experiments behind us, we set off to generate large numbers of populations and progeny in earnest, and this gives an idea of how we made those crosses. We bulked different Hyperethrum genotypes. We didn't want to use just one accession because we wanted to avoid the possibility of inbreeding depression in later generations. And we'd cross those with an array of cultivars. In this case, this is the red group of varieties. We're looking for, strong pinks or reds out of this cross. We also used yellow, purple, whites, so forth. And we grew those out in quantities with the thought that we could possibly make some of that first generation material might be worthy of introduction, but then to take that to the F2 generation through SIB mating strategies, either full or half SIB. And this shows the crosses were made in 2005. In 2008, we had them growing out in field rows. They all have kind of the same look, pretty dense. This is in the open sun. They do pretty well. You can see flower buds on them. By year 4, most of them had produced flowers to evaluate. And of course, they were spending the winter outside. So we were getting a performance and an ornamental evaluation within 4 years by 2009. And here it shows you some of the selections we made out of those thousands based on ornamental value and performance. So we were reasonably satisfied since one of the parents was white. We were happy with what we were seeing in terms of color saturation. Oftentimes, too, you get this nice patterning. Gardeners call them blotches, but of course bees know them as pollination guides.
And are any of those that we're seeing now named cultivars?
Whoops. On this list, actually no, but I will finish up with those. I'll show you. Okay, in fact, I'm going to peel off from the breeding program at this point and pick it up at the end of the talk and detour a bit on some stress testing we've done. So I've been talking so far really about transferring resistance genes to progeny, and in pathology the operating principle is called the disease triangle. I know many of you have probably been introduced to this. So the disease symptoms we see in outdoor conditions is really the interaction between the host plant, the pathogen, and the environment. And the environment can have a huge effect on moderating the outcomes. And typically, stress environmental conditions reduce resistance. That's a phenomenon that's called predisposition. So if you look at abiotic stresses like extremes of moisture or temperature, salinity, ozone, those are all things that can somehow change host physiology in a way that weakens its defense response. And then in particular, I was interested in looking at flooding and heat, because those are two traits that deal immediately with our desire to move these plants south, and also with really the heavy kind of substandard soil conditions you find in most landscapes. So in 2011, we conducted a field flooding trial. We put out an array of genotypes, some unknowns, as well as some controls, both susceptible and resistant checks. And this was an area where we suspected the pathogen was present in our fields. The idea initially was to flood the area by pumping in water from our pond, but it turned out to be the wettest year on record in Northeast Ohio, so Mother Nature took over our treatment plan, and we had conditions like shown there on the left repeatedly throughout the season, probably on 7 or 8 occasions, where we would flood, and then over the course of a day or two it would drain. So by the end of one season, then there were clear, most things were looking pretty bad, from outright dead to oftentimes chlorosis is a sign of stress, of root rot damage. But what really stood out was how well Hyperethrum performed under those conditions. That's shown here. And when we did it, so we harvested, all the dead plants got root scores of 5, and then we harvested the survivors and calculated the values for those. And this shows the breakdown. We had groups of susceptible plants, resistant benchmarks, 3 of those are resistant plants that don't have hyperethrum in them, some experimental hybrids with unknown resistance levels, and then the species itself at the bottom. And on the left here, this column shows disease scores under non-flooded conditions, and these were determined in previous experiments before the field trial. So, our resistant group here and here had roots values of about 2, which is equivalent to just damage to fine roots, but nothing further. Under flooded conditions, some clear changes occurred. Everything was more susceptible under stress, but in particular, the resistant benchmarks almost doubled from their root scores. So that's equivalent to from going from just having fine root damage to damage up in the crown region. So these plants were nearly dead. In contrast, hyperethrum, although the values went up in root scores, only about 30%. And the hybrids of hyperethrum did quite well overall. They weren't significantly different from the parent species in terms of root score values. And we're attributing these results not to flooding stress directly, but to pathogens. And hypoxia is a possibility. Typically, it doesn't lead to plant mortality. And rhododendrons can handle about 48 hours of flooding with no issue. Plus, the plants really, the sequence of mortality in the field really followed our expectations based on our benchmarks, so that the susceptible plants died right away, resistant plants held on longer. And then to kind of close out that question, we also isolated Phytophthora from necrotic regions of the field plants. And so that's done by, in this case, we took I think pieces of stem near the crown region, grew them out on semi-selective media, which will screen for Phytophtheras, but not at the genus level, but not at the species level. Phytophthora cinnamoni, though, does have kind of an unusual morphology. It's coral-like or coralloid, so that's another indication of a potential match. And then finally, we had those samples sent to Ohio State, and they did a sequence analysis against there's a diagnostic storage protein in Phytophthora cinnamoni, and the match was 96 to 100%. So we're fairly certain that really flood, the Phytophthora was causing the symptoms, and that it was that some of the effects we were seeing were aided and abetted by the flooding treatment. So in conclusions from the flooding trials, that it's less predisposed to disease than other resistant rhododendrons. The trait's heritable because the F1s did statistically, they did as well as the resistant parent. And this is good in the sense that in terms of a product development, because we could potentially develop rhododendrons that can withstand those poor soils, heavy soils found around in home landscapes. We also looked at heat stress, and this is another key question, being that are these plants that are adapted to warm climates, like hyperethrum, less predisposed to disease under high temperature stress? And to do these kinds of experiments, we chose a pulse treatment method where you could expose tissue, in this case roots, to heat, remove that stress, then inoculate it and include uninoculated controls, and then subsequently measure the level of resistance in those treated plants. And we use large water baths, such as this one here, to apply heat. We grow either single plants or seedlings in pots, and then drop those pots into a poly bag and immerse it in the water bath for treatment. And our treatment temperatures range from sort of room temperature up to about 42 Celsius. Typically, the experiment was done 1 hour to target in the bath and then 1 hour at the target temperature. And these are some data we just got this winter comparing Asian species for the response to heat stress. And I just have summary means for these. I haven't run the stats yet, but basically we have red. So these are root rot scores plotted against days after inoculation, and we're comparing on the bars the controls with or without heat, but no Phytophthora, and then with or without heat plus Phytophthora being the green bars, and the dark green is the heat treatment. So one thing to point out in the controls is that there's no lethal effect of this heat stress on the plants. And in this case, this is a species from Central Asia called Panicum. It's a susceptible plant. It was really our control, and we reached almost complete mortality within a week and a half of inoculation in both the non-stress and the heat treatment. No surprises there. I mean, it's hard to make a very susceptible plant more susceptible. In contrast, though, we looked at 2 Asian species that are resistant, one from Japan called Kiskii, which is not considered to be heat tolerant, and then our heat tolerant Hyperethrum. And in those cases, the heat stress in Kiskii, the dark green, clearly elevated the disease scores, especially in the early part of the time course. Again, I don't have a test yet on significance, but it looks like it could be. And Hyperethrum, except for the very first sampling date, which could be sampling error, all subsequent dates, the heat stress plants showed the same as or lower root scores than the non-stress ones. So it seems to suggest that Hyperethrum is not predisposed to disease by higher soil temperatures, much in the way that it was less predisposed by flooding. So those are good 2 attributes, in addition to resistance per se. Another study was more ecological. So we wanted to know whether in natural populations are warm adapted ecotypes more disease tolerant following heat stress than their cold tolerant relatives. And to do this, we switched to a species called Rhododendron minus, which is native to the eastern US. And it grows from high elevation North Carolina down to within 50 miles of the Gulf Coast, so in the Florida Panhandle and southern Alabama, for instance. And we've been collecting those over the years, putting together, trying to capture really a range-wide collection of this species. And we have compared two populations so far. The one here at elevation 4,000 feet in North Carolina, and one at low elevation in southern Georgia called Providence Canyon. So this is the shrub here at the top of Hawksbill Mountain, and you'd find the Rhododendron minus at the bottom of this canyon along the riverbed in Providence Canyon. So we did the same kinds of disease progress plots, same kind of heat treatments that I described earlier. Temperatures were a little bit different. I'm showing you results at 26 and at 42 Celsius. We also did them at 34 and 50 degrees Celsius. Looking at mortality rates over time. So at 26 degrees, and the blue is the northern high elevation population, and the red is the southern low. And these are the controls down here. So heat treated, but not inoculated. Again, there's no, heat treatment appears to be sublethal. And at 26, they're basically the same, statistically the same response. Both species are highly susceptible to Phytophthora, so there's mortality within almost complete within under 2 weeks from inoculation. And you would expect that in the case of a natural population that's being challenged with an invasive pathogen. It's never seen it before. At 42 Celsius, I mean, there's a high degree of mortality and ultimately in both populations, but the rate of disease development or mortality in the southern population is much slower. And looking at areas under the curve, here that turns out to be a statistically significant difference. So there is a transient effect anyway. The heat stress appears to be initiating some sort of defense response in the warm climate ecotypes that's not found in the cold ones. So that I thought was a fairly novel result, and we want to see if we can generalize those data looking at a larger sample of populations. Still, the question of root damage is there. You know it's sublethal, but you don't know if there is something going on at 42 Celsius. That's pretty warm. So we, and that could confound the interpretation of results. So we did some viability assays on both populations using TTC staining, a vital stain that will turn cells pink as it's reduced by mitochondria. And you can elute that stain and measure it with a spectrophotometer. So here we're showing absorbance standardized over dry weight, milligrams dry weight for both southern low, northern high populations. And there is no significant difference in the viability estimated that way. So we feel that within that 26 to 42 Celsius window, root viability is not an issue, and that's a good range of temperatures to continue working in. At 50 Celsius, I mentioned, we had done a screen there, and there actually was probably 50% loss of viability in those tissues, a bit too extreme. Well, we don't really understand in rhododendrons what's going on at the physiological level, but there are reports, there's increasingly reports from other systems, notably Arabidopsis and tomato, of how predisposition might function. And it really involves sort of antagonistic metabolisms. So you start with a resistant plant, given an abiotic stress, that kicks in the hormone abscisic acid, which is it's a very common upregulated stress-related hormone. But increased ABA can cause a inhibition of defense signaling pathways that are specifically those mediated by jasmonic acid or salicylic acid. So if there is then a pathogen challenge during or subsequent to that stress, then the defense system of the plant is lowered, and you can change phenotypes from resistance to susceptible. That's kind of the consensus model. There's certainly lots of variations on that, and we know. And then what I feel from my work, since we've identified genotypes that are less predisposed, that clearly there's probably involved some different regulation at the gene level. And with the advent of less expensive genomic analyses, metabolomics, comparative transcriptome analysis, these would be perhaps fruitful studies for the future if you were predisposed to that. All right, well, finally back to the breeding program. Since we wanted to develop heat-tolerant rhododendrons, the obvious thing was to put them in the south and see how they did. For this work, we found a industry partner with plant development services based in near Mobile, Alabama. They have a brand called Southern Living Plant Collection, which is quite popular. And PDSI really, they're not a nursery per se. They work between breeders like me and the marketplace. They take care of the development aspects of the plant. Those are services that we don't have in-house at Holden. And the test site was in southern Louisiana, Independence. We had put in over 165 selections that we made in Madison initially, and put out a replicated field trial. The plants were shaded, I think, for the first half of the first summer, and subsequently have been out in the open. And this is now the beginning of year 3 of the trial. So they get fairly strong afternoon sun. And based on the results so far, there are really about 20% or maybe 30 out of the 165 selections that we put in have really performed well. That is, we're at 100% survivorship, and then the surviving plants themselves are vigorous growers. They have good leaf quality, and they're producing good flowers, such as the one shown here. And this past March, I was down there while they were in bloom, the first time I'd been there in bloom, and we selected 3 for introduction. Those are the ones shown here. So 2 pinks, one kind of a clear pink, the other more of a rose pink. Some of these patterns, these flares and blotches. This one has a frilled margin to the petal, as does this. There's another nice feature where the petals, the backs of the petals will be darkly pigmented and the face will be lighter, so that creates kind of a bicolor effect. And these little nuances do make a difference. It makes the plant fancier. And we hope that these can compete with what's already kind of been established as the current standard for rhododendrons. And of course, in the south, they will have no competition because there are no rhododendrons yet that have been developed. So these will go into tissue culture. This one already is, and then subsequently out to growers. We can glean some extra information from that field trial by, we want to confirm the presence of Phytophthora, just as we did in Madison, Ohio, by isolating and sequencing the pathogen, just to prove that that's what's causing it. It is an old nursery site, and they apparently grew a lot of azaleas and camellias there, and those are both hosts for Phytophthora. And because nurseries are constantly moving plants in and out of their inventory, and Phytophthora travels in soil, it's quite likely that it's there. Certainly the symptoms are consistent with it. And then it would be good to go back and look at both the top and bottom classes of performers. We have the original mother plants still in Ohio, and so we can go back to them and do tests for resistance under controlled conditions and confirm that they are respectively resistant or susceptible, and that will really close the circle on this notion that disease resistance is a key trait to surviving in a warm climate like that with high disease pressure. Well, at Holden, we always talk about being about more than just a pretty plant, because we're a serious institution on a serious mission. But even though the scope of my project is fairly narrow, I've tried to move it in a direction that's topical, that addresses the issues that knowledgeable scientists are concerned about in these days and age. And so the issue of sustainability is addressed because resistance will probably result in reduced pesticide use, typically fungicide drenches that are used in nurseries. Plants are broadly adaptable. I think my goal of covering zones 5 to 9 has probably been modified by the past 2 winters, and I think we will release these marketed to zones 6 to 9, although you can, many zone 5 winters it will do fine, just as it has done well in Ohio up until recently. We'll call them climate change ready. Certainly they're positioned for global warming. And warming is not just an issue in terms of stress on plants, but most predictions state that disease pressure will also increase under those conditions. And there's an increased chance with warmer climates that pathogens like Phytophthora could naturalize in our northern forests. And finally, it's a good example of a solution-based use of biodiversity, showing how we can tap a species like Hyperethrum to address a real stubborn, problematic issue like root rot disease. Finally, I'd just like to thank some of my staff. Jing Wang is a research associate who works year-round with me. And Hillary Wright has been a seasonal. One of our volunteers, Susan Hanna. And my collaboration with PDSI is through Buddy Lee, shown here on the right, who's the Director of Plant Innovations for PDSI and also an accomplished ornamental breeder in his own right. And finally, a few sources of funding, not to mention my major underwriter, which is the Holden Arboretum. And that's it.
Okay, we can have the lights on.
You had mentioned you had like maybe 10 genotypes of Hyperethrum that were used to prevent input depression. Were those selected for superior performance in terms of disease resistance from within a broader group?
They were. Although there is some structure to those, they were. I didn't have any Hyperethrum plants when I started. It wasn't in our collection. But there's an outfit on the West Coast called the Rhododendron Species Foundation. They're in Tacoma, Washington. And they have a pretty good world collection, including several Hyperethrum. So they made some crosses of those accessions, their accessions. They sent me seed, and then I screened that for resistance and took out the survivors that were really the most vigorous. We didn't know about their flower attributes at the time we selected those parents. They're mostly smaller, but there are some different. Some seem to have larger flowers with maybe more substance to grow than others. Yes?
Do any of the new varieties have ornamentally interesting fruit on them?
No, they're not really known for showy fruit. They're green turning brown. Yes?
I was going to ask if you have any idea what the resistance is.
No, I don't really. I mean, it's polygenic. It's not an R gene, so it's probably multi-level. It could involve phytoalexins. We think that there's an age-responsive difference. I didn't show that we didn't discuss it on the heritability plot, but the offspring scores were the whole, they had shifted up in value, so they were more susceptible than parents as a group because we were measuring 6-month-old offspring versus physiologically mature parents. So there could be something there. Yeah.
So when you name new varieties, how are we going to know? It's going to be that each had different famous cities.
Geographic names?
Right. And so what are you going to do so that periodically we're going to know that it's your variety?
Well, I guess a lot of it may be somewhat out of my hands. I think PDSI will probably have their own ideas. And they're really, they're the marketeers. I think they do a lot of that, and maybe they have a sense of what contains with so we haven't quite got to that point of discussions. But we sort of have we sort of do what I call Leach classic hybrids that we're introducing. We're sticking the name Holden in front of this. Holden is solar flare, so forth, things like that. So you will know those. Those wouldn't be for the South, they would be for the traditional market.
Well, I think we're out of time, but again, I want to thank you, Steve, for traveling to Ithaca and sharing your research with us.
You're welcome. Thank you for coming.
This has been a production of Cornell University, on the web at cornell.edu.